So, welcome. Whoa. I'll talk from about here. So, ordinarily they have you read a bunch of housekeeping and ground rules. I think the ground rules for this meeting is basically participation is required. So, get out your phones, your computers, whatever you need to be able to participate. You know, I hope we have a lively conversation and you all take a bit of activity back with you when you leave, and then you give our group some work to do moving forward. So this session is put on by the Core Rigor and Reproducibility Committee here, so you can read the mission, and actually I cannot. But this is really a session that's put on by the committee. It's no one person that can do all of this, and so. You know, you'll see all the names as we go through this. But what drives this for me in terms of, you know, how do we support rigor and reproducibility at our institutions was this very surprising survey result that I saw. And basically what this came down to was a situation where if your institution actively supports rigor within the core, and by actively, it's a pretty low bar, right? Do you have a statement, right, concerning rigor and reproducibility at the institutional level, and the core has a statement on their website? You know, it's on the order of 80%. Of those groups are actively working with faculty in terms of working to improve the quality of research within the core. And importantly to me, it's at the grant stage, right? So this is the stage where cores are constantly saying, you know, they want to be involved, right? So, you know, the number of times you hear, you know, well, we could have done a great study if you just would have funded enough samples, right? Or some controls, right? Having that statement on rigor and reproducibility, right, means that you're more likely to have that conversation. On the other side, if you don't have that statement and you don't have it at the institutional level, it's sub 10% of those folks are actively involved with faculty and experiment design. And you know, you can kind of see the numbers, right? There's 40 cores or 45 cores, right, that answered no to both of those questions in the survey, and you know, on the order of 12 that answered yes to both of those questions. And so to me, what that means is it must be a hard conversation to have, right? So we're not at birds and the bees difficult, right? But it's a conversation that appears that people are avoiding, you know, and I'll feel free to come up with reasons for it. But I think that it's important to go back with the idea of we're going to initiate this conversation in a place where it's comfortable for you to have that conversation, right? So don't make it, you know, the most difficult starting place. Make it somewhere where you feel like you can speak from a position of authority, and something that will impact your research. And so our group thought, you know, how can we leverage this meeting You know, and with this idea of how do cores initiate the conversation. And so what we wanted to do was identify opportunities to initiate these conversations. We wanted to make it feel like, from your standpoint, it's a good thing, right, as opposed to a scary thing. And then identify, you know, why aren't we having these conversations now? So that you can figure out, you know, how do we make first steps and have these conversations. And then rightfully, you should come back to ABRF and say, hey, this is what we need from ABRF to make this process easier. And so as we talked within our committee, we came up with the following ideas. And I'm going to briefly go through these ideas, hopefully make sure we didn't miss any. And then we're going to play a game of musical chairs where you find your tribe and maybe move some tables together. Very excited about the level of participation here. And then the idea is to take about a half an hour to talk at your table and then have each table report back out on what you've discovered. And really the key idea is, you know, what action item do you take back to your institution and what action item does ABRF need to take? So that's the deliverable on the tail end of this. So I'll go through. So first one is data management and sharing, and Sheena is hosting that one. You know, and this is a daunting task, right? So you know, if you're waiting for the perfect solution, you can see that, you know, how many steps there are in this process, and I feel like sometimes that makes it hard to get started, but incredibly important. So next is figuring out just what happened in terms of how experiments were run. And so we know, right, number one sign of a failed organization is a name change. So I can say that because I came and joined a core and 2 years later changed the name. That makes it very difficult to find information, especially in older papers. And so we know how to solve that, right? We solve it through the use of persistent identifiers, but now we have lots of persistent identifiers. And so, table over here. So next one is methods documentation. So I'm going to host that one, not that I'm an expert in methods documentation, but I got the table, and really what this study in eLife showed was, you know, over half of the studies that they tried to reproduce in this study, they couldn't do it from the start. They had to reach back out to the authors to figure it out. And disappointingly, a lot of those authors were not helpful in the process. And so as a core, you know best what methods need to be documented and what level of detail is required. So that'll be this table here. The next step is Francis and Katja, corner over here. This is something that is widely variable in terms of cores and how much they participate. But you are the experts when it comes to reviewing data quality and experimental design, right? And so, you know, there is definitely a seat at the table in terms of peer-reviewed journals and making sure that the quality of the data and the experiment is as expected.
Can I say something? This is an old slide. So there will be a QR code and you can see what actually we decided to cover today with your help.
Okay. So some of what we need to do is just create a culture where we set a high bar for the level of work that we're going to do, right? And so I talk about when I came to join an analytical chemistry lab, the staff would walk around and say, how's the instrument working? Right, which doesn't set a culture of, we know the instrument is working fine. And so Chris is going to host creating that culture, both within your groups and within your user community. You might be wondering, should your lab have an accreditation or a certification, right? And when does it make sense to go after that? And will it help you either generate business or make sure that you have the systems in place to conduct your research? So Kevin is hosting that one. And Nicole, thank you. And one thing I want to ask before we break up is, do you think we missed any important topics? So we have enough tables. So, go ahead.
I think we'll join over here. I think there's creating a culture now.
Okay. Okay, so now what we want you to do is get up, find your tribe. Okay, we're going to spend about a half an hour just open discussions within the table. Okay, there's going to be some more materials available to you. So if you scan that QR code, your table may have some extra information. And then at the end, the table leaders will present out.
Sheena let me do a presentation to all of you. Oh, remember? I don't remember. I do now. You're up here. Oh, I know. Like alert. So are you going to figure it out? You're walking into? You actually would have like a And I think cores are a really great place. I would also advocate that core directors and staff
describe it in the same way. So I think that's something that's coming in. But I think it's an example of one of the cases where we use the text is and you can hear it if you have a repro where your curve is so a new version of the same. Yeah. And a work would be like find experience from someone who's in that situation. So it's like a repository of
So between 5 and 10 minutes, so start thinking about distilling some thoughts down to action items. Okay, so it's time to close up with your final bits of things that we're going to talk about here and report out. So much good conversation. So I'm going to take the, my, it's, well, whatever. I'm going to go first. So our table was working on methods documentation, and what we had come to the conclusion of is the cores are actually doing a lot of it already. We don't really know if it's going into the publications or not because the time delay and the bandwidth needed to review publications is basically makes post-market surveillance an impossibility. And so our group decided to focus still on the front end of this. The big issues we came up with are the perceived cost of going through the methods documentation and review, and time. So documenting what you've done, the review and the complexity of different procedures. But the cores are still the right people to generate these, right? So if we think it's a lot of work for a core to do it, imagine a faculty that has to do it for 5 cores. And so, we thought that that's a reason that's good for the core to do it, right? There's value add to the faculty. We're going to save them time. It's a publication opportunity for core staff, right? So that's a chance to build your resume. And I forgot what save means. The one thing that I think we do poorly that the cores could take back right away is to advertise their methods in context with rigor and reproducibility. So if you put your methods in your training documents or you put them as posted protocols on your website, right, just adding that little bit of link to why you put it there will be value added. We did have a lot of discussions about providing the method development as part of the consultation process and including that in the base rate of your cost, right? So you're going to do it no matter what. Don't charge separate and have people thinking about avoiding. The publication tools that we identified, so your own website is okay, but bioprotocols and protocols.io would be better in terms of your authority and career development. And then the request from ABRF that we saw moving forward was that with the adoption of electronic notebooks, right, for how cores should use and integrate electronic notebooks into their process is a generic problem that's going to be coming around to everyone. And so that's all we had. Who wants to go next?
So we talked about creating a culture of rigor and reproducibility and transparency. And great conversation. And one thing I'll say, and not to throw anybody under the bus, nobody at the table had a rigor and reproducibility statement that they operate with or have on their website. And I think that's probably universally true, and that's not necessarily a bad thing. It's just the first step is admitting we have a problem, right? And so that's kind of what we did at the table, a lot of confession here today, so it was good. So we have a template that's available, and I think it's on the ABRF website. It was in the paper that Andy talked about a little bit earlier. Happy to provide it to anybody. You can fill it out for your core. It's very easy and straightforward. The point was made that just having a statement is not enough, right? You can put everything you want on the website, and they will ignore it all day long. And so the idea is, and what we all agreed was a good idea, was to sit down with them during the initial consultation and actually go over this statement. This is how we operate. If you're not happy operating this way, then we're going to say no. And that was one of the other things we talked about at the table, is when is it okay to say no? And a couple core directors have no problem saying no. A lot of folks eventually get to yes, even though they don't want to spend the money in that way. And so what we emphasized was that's the first step in rigor as a core, right? Is saying no or saying yes, but. We need to have a deeper conversation about your controls or about how this experiment is designed. And so the other thing, in addition to posting on the website a statement, is to put signs in your core, visuals, posters on the walls. You know, don't underestimate the power of an image beside your microscope, right? It's worth 1,000 words. If you put your RRT statement up there as well, it's hard for them to ignore it. Use training sessions. So some core directors and managers do training for their customers. During the training, talk about rigor and reproducibility. Now, that's an easy thing to add, you know, as a couple of slides, and you've got a captive audience. During the initial consultation, of course, talk about experimental design, but also build in that idea of rigor and reproducibility from start to finish, right? When you get your data, this is what all the steps it's gone through to there, and you can trust it. As far as what ABRF can do, education. And someone made the suggestion that as part of the town halls that we have, that once a quarter it's focused on rigor and reproducibility and transparency, which I think is a great idea. For CCoRRe, I think that's a heavy lift to have something every quarter, but it's definitely worth talking about it and something that we should consider. Did I cover it all? All right.
Katerina?
Hello, hi. So we spoke about persistent identifiers, and most people that came to the table were interested in the persistent identifiers for the core facility, so the use of RRID. So we discussed for core facilities, so we discussed about how to make it maybe more attractive and how to incentivize its use, and we definitely discussed about, for example, the work that Chris and, sorry, I can't remember your name, Kara did to link, to be able to actually automatically get information about the publication as a really important key piece of incentivizing the use for the institution, for the core. And then we transitioned, so everybody agreed that that is really an important thing to do. And one thing that was mentioned about how it could actually be maybe incentivized and linked to actually how much money each core brings in is its impact on revenue for the university is asking the sponsor program office in your university to ask, as part of the process of signing off on a grant, which core facility are you going to use, and so that people can actually, so that this information is directly linked to a grant, and using RID in that case it's clearly a good idea. Then we transitioned to thinking about RRID for instruments, and you know, that is a heavier lift. First of all, because, first of all, RRID for instruments are linked to actually the instrument model, not the instrument serial number. So then we're, you know, the idea is there be any value in having a PID for the actual individual instrument versus, in addition, to the model, and you know, how hard or difficult is that, and what added value it could have for the core facility managers and everybody involved. And we discussed about how if we link some of the, you know, as we're thinking about methods reporting, having a full description of an instrument and having associated with a specific PID inst for that instrument, it is actually could be make it much easier for the PI to add value to their methods. Then that could be an incentive for PIs to use the whole instrument, the whole idea of and then that could also end up being a way of incentivizing the use of RRID for cores. Also, it would be a way for cores to better advertise what they have in their facilities and draw more customers in, because now scientists can actually compare apple and apples if we're using community-defined guidelines for describing the instrument. So we didn't come up with any solutions, but we had a lot of different and important discussions, and I think it will be important to carry this conversation forward. Did I forget anything?
Request from ABRF.
Oh, I think it's actually a good idea to especially with what was developed at UNC, start actually maybe being a little bit more proactive in promoting the use of RRIDs for core facilities, because now we can actually say that there is much added value, because you can track the publication much easier. And then, if we then link to the grants, that is another important aspect. So.
Okay, Nicole.
Our table talked about lab certifications. We started our discussion off by really just defining differences between certification and accreditation, so we're all on the same page. So the number one thing is, like, why is this good for core staff? I think we all had a nice light discussion on it. Just generally increases awareness of the value, the recognition, and the accountability in which the Core is paying attention to the services that they might be providing to their investigators. So overall, we thought that it was a good value. But the challenges with accreditation and certifications are numerous. One is, how do we get started? Identifying the type of certification that best fits the mission for that lab, and finding an agency that provides the accreditation for a given certificate. We talked how there's some disciplinary areas within core facilities that have certification processes and others that just don't have it yet. So how do you get those conversations going? The cost of certification varies from hundreds to thousands of dollars based on institution or lab size and the accreditation label and scale that you want to bring it up to. Another is time. It varies within the type and certification level that you're going for. Some don't require a lot of time. Some require a tremendous amount of time and information that you have to submit in order to acquire that certification. Personnel also varies, as some may require the hiring of additional personnel just to oversee the certification program, and some you don't need personnel to oversee it. It just really depends. Infrastructure, every institution structures and functions differently, right? We're all our own different little cells. I'm going to my biology days, structure and function. Typically a significant lift to some of them that might be more centralized have better processes in place, because you have to be able to write and update SOPs, data records, maintain instrumentation, maintenance, lab environment, safety measures, et cetera. We just had a healthy discussion on service contract earlier and how to centralize some of that information and the challenges we all face. Everybody's different. Other challenges are, that came up just in our discussion is the service overlap. Core facilities, I know in our institution and some others as well, we have some that share space and share instruments. So how do you do certifications when you have a clinical and a research assay sharing an instrument, or staff sharing an instrument? That's kind of like a balance that is a struggle with. In our policy, we have to certify the entire lab, and that's a cost to the researchers, and so we have to pass it on sometimes. And then just generally, also another challenge is identifying just space in general. Like, how do you certify the space? Because we had a conversation on square footage, like when you have to update things, and how do you negotiate with your institution to get that kind of things corrected for, and cost. The other thing that we came up with is, how can ABRF help? Number one is, reading my chicken scratch here. Hit 43, my eyes are going south. What can you, wait. Oh my God, this is terrible. So ABRF can put forth like a how-to guide, kind of like a quick decision tree. So what certifications can you start with, and how much time and investment do those various certifications require? So people can probably hit the low-hanging fruit easier, and then if they have time, they can divest more into the fruit that's at the top of the tree that requires more time, more investment. Just start simple. One of the things that we thought starting simply is migraine lab, you know, get some sustainability certification or move into your area of discipline. And then identifying people within ABRF that have been through this process and really putting their names out there. These are people that have done this, they know how to do it, they can help you contact these individuals, and they'll help kind of rally you around that. And then just an overall, within that decision tree, a how-to guide, like these types of certifications, brief high-level outline, require this much effort, work, this type of documentation, this type of paperwork, et cetera. Anything I missed? I can't read my chicken scratch, so, okay. So that's us.
Okay, so our group was data management and sharing, and we had a lot of good discussion in terms of the role of the core. I think everybody agrees that cores are data generators, that they generate a significant amount of data on any campus, but we all agree that every institution is different, and so there are institutions who have data governance, there are those who don't. Every core is different. Every type of data that cores generate is different. But what we came up with was the fact that every core in generating their data does in fact have a data management plan. And so that information in terms of what each core does in terms of their type of data can be shared with investigators at their institution for the creation of their data management plan. So we had come up with a template in CCoRRe that would be beneficial to share as best practice, and to at least start getting additional input in terms of does this really meet the needs of ABRF member cores. However, it shouldn't just end there. It also needs to be elevated to NIH, because as Yvette has pointed out, there is significant difference from IC to IC to IC. And so, in fact, we have to get this information up to NIH in terms of where does the data really start? The data starts in the cores, okay? So that was one thing. The other was best practices in terms of what really constitutes a data package. And so, for instance, in terms of single cell characterization, in terms of some of the spatial transcriptomics, you know, what constitutes best practices in terms of the data that really we're all involved in, which are these multidisciplinary, transdisciplinary projects. The other is in terms of the data cost. Everybody, you know, how much data needs to be stored? Are there really standards? You know, like again, getting back to the core, how much data does a core hold on to for any project, for how long? You know, is it 6 months? Is it 2 years? Is it 3 years? But it gets into the cost of data storage and also the amount of storage. And so, again, every institution is different, you know? Everybody has a different rate. And so, yes, there is money available for data storage, but it has to be coordinated between the cores, between the grant writers. And so this also highlighted the fact that every institution is not necessarily creating a transparent system where the information from cores to investigators, and this gets back to what Chris was talking about, which is at the beginning of a project, all of this needs to be discussed in terms of, okay, ultimately, what is the deliverable, what is expected, what are the aims, et cetera. So again, data management and sharing all fits into that. And, finally, in terms of what is really needed, we're all asking this question, but I think this is where ABRF can bring value in terms of ultimately coming up with best practices to share with the cores to contribute to best practices for data management and sharing. So I'll leave it with that. Did I cover everything? Yeah, there's so much.
Okay, so our table discussed, it's called publication review, but it's very much focused on the contribution that our community as technology experts can actually do to elevate the rigor, the reproducibility, and the transparency of what actually gets out there, right? So these discoveries are wonderful and they're cutting edge and they move the field forward, but if you're unable to reproduce them, then their value really gets undermined. So we answered a few questions. The first one was, what do you think is the most important outcome of peer reviewing done by cores? Whether you be a core director or a core staff member, your title is irrelevant because as long as you have the expertise. And so it was to make sure that the information is accurately recorded, to ensure that the experiments are reproducible. And also it should eliminate or at least minimize conflict of interest. We all know that scientific reviewers often have a conflict of interest with the manuscripts that they're reviewing because it is in their field. We should be relatively agnostic most of the time because not too often we are authors on publications. And what are the top the Q2 was what are the top factors or reasons? One, a core facility person would accept a review. I added one, sorry, while I was rewriting all of this. One is altruism. We are part of the scientific community. We make an important contribution and we should take, as Katja has said before, we should take our seat at the table. But we have time constraints, and so what's in it for me? Why should I do this? Why should I spend time not building my core, not managing my core, not pushing the technology forward? And one is we can add it to our CVs. There are institutions that will recognize this contribution as bringing value to the core director. The other thing is you learn about what other people do, right? You learn about changes to the technology, evolution of the technology, different applications of the technology, because each institution does different things. And so when you learn that, you bring that, you not only build yourself professionally and do some professional development, you bring it back to your researchers. And that is part of our goal, is to expand what we make available to our researchers. So what are some of the reasons you might decline this? You know, someone comes and asks you for a review, and one is time. I mean, we are all, our bandwidth is very small, right? To do something outside what we are charged to do for our researchers. Sometimes there is a conflict of interest. Sometimes it's a bad match, so you get asked to do something, and maybe you can do half of it, but you can't do all of it. Right? So I've been asked to review things and I know nothing about bioinformatics, right? And so I say, well, you know, I can review this part, but I can't review that part. And some people, and this is one thing that CCoRRe really wants to, CCoRRe wants to address, is people not feeling that they have the expertise to do this, right? So, you know, some of us reviewed manuscripts if we were a PI before or during our postdoctoral studies, or maybe some of us never reviewed an article. You are the experts, and how can CCoRRe help you overcome that barrier so that you feel qualified to do so? The fourth question was, what type of training can ABRF provide you as a reviewer, whether you've reviewed or not, to actually bring the caliber, hold the level of reviews that ABRF reviewers can provide to a nice high standard, right? And so some of the communities, like the animal community and the flow community and genomics, kind of have some guidelines already. We could make our own, add to them for the other technologies, create them, and provide them to the reviewers, so we are providing a uniform quality of review to the scientific community. And I think that's what I have. Katya has some more points.
Yeah, so first of all, I want to thank David, Galina, Gordon, Tanya, Barry, and of course Francis and Katya, because I'm going to read the working group summary, and they all approve this message. And so we have volunteered them to become part of a task force. We collected all of the emails, and we're going to build something out of this, because this is what needs to be done. We can talk about this ad nauseum, and if we don't act upon it, it won't go anywhere. So we, as a group here, champion of rigor, reproducibility, and transparency when it comes to our engaging into scientific publishing. We recognize the need for the course to be part of the publishing activity as reviewers and authors, co-authors or whatever, to enhance, to enhance the quality of science that is out there, as Francis wonderfully put it. I cannot say it as well as you do. We recognize that there is a need to create or increase the reviewers' pool from the ABRF. And using the ABRF membership pool, this is rich. This is a rich pool of people that are capable of reviewing. They may not know it yet, but they are. We are the expert. And maybe Francis can talk about something at the end of my little summary here. But you will discover that you are wanted by the editors. So we recognize the need to ensure that the reviewer in that pool are well trained. We need the training. We're not sure what can we contribute to this. So please tell us how we can do a better job as reviewing. And then we recommend as a group here, and we are going to be participating, and we support initiatives that will provide formal training for us guys, maybe during ABRF or during the year. And we recognize there is a need to find ways to reward reviewers. You know, when you're a scientist, you're rewarded by reviewing, because then it allows you to be in the pool of people that they can promote to assistant professor or full professor. We don't have such thing in the core curriculum for advancing our science. So we need to work on that together. And also it will also allow us to maintain motivation to do more review. Now, Francis, would you like to talk about?
You have 1 minute.
1 minute, I promise. So the AACR has come to ABRF through CCoRRe seeking technical reviewers. We are currently in the process of trying to create a path forward for that. So if anyone is interested in this room, or your colleagues are interested in being reviewers, please let us know.
So this is for just the methodology part.
Yes, this is a technical review of the article, so that would be methods and figures.
For AACR journals?
For AACR journals. And so this is they're creating a platform through which we would submit our reviews and stuff like that. And so we're going to start off with genomics to kind of develop this pathway.
One more. When an editor contacts you and you think you don't have the expertise, tell them. And then they may say, but I contacted you because I knew you're the champion of rigor for genomics. And they say, don't worry about it. I have other people to judge the science of that paper. So talk to the editor, talk to the person. Don't say no because you're like scared that you're not at the level that they need. Tell them what they need from you. And this is what the AACR journal needs from us, that technical ability to realize, you know, this is good or this is crap, and they should not publish it. So.
Okay. So thank you all for your participation here. You know, I heard 2 things over and over again. So one is volunteer. You have to put yourself out there, right? Whether it's with your administration in terms of things that you need to be defined or to review or to help others along the path. And the other is to incorporate rigor into the things that you're already doing, right? So if you're redoing your website, make sure there's a rigor statement. While you're training, there's an opportunity, right? And make sure people understand the rigor part of it. So in closing, I wanted to say congratulations to the group that formed this committee. So Kevin, Katja, Francis, and Sheena. This work is being recognized by the NIH, and so the initial folks involved in creating CCoRRe have won the Rigor Champions Prize for 2023. So congratulations to you, and thank you. That's it. You may go. Enjoy.
You can't get what you never had.